We'd like to welcome Dr. Andrew Kulmatiski. He's a new associate professor in the College of Natural Resources at USU. He's received degrees from Colby College, the Yale School of Forestry and Environmental Studies, and Utah State University. Andrew studies plant-soil interactions with the expectation that improved understanding of below-ground processes will improve natural resources management. So can we welcome Dr. Andrew Kulmatiski?
Thanks. I think I saw my department head in here. Sounds like I just got a promotion. I'm actually an assistant professor, but maybe Mike put in a good word. I'm glad to be the warm-up band here for Jerry Franklin. I figure I can either do a bad job and make him look good, or I can do a good job and warm things up for him. So it's win-win for me. I'm going to change gears. I'm going to talk today about some pretty basic plant biology and an embarrassingly simple question, and that's where do plants get their water from? It's amazing, but we still don't really have a good picture of where trees, grasses, and shrubs are getting their water from. And there's a lot of reasons to be interested in it. This is pretty fundamental, basic biology. You have water, especially in semi-arid systems. A lot of it hits leaf surfaces and then evaporates even before plants can use it. Some makes it into shallow soils and is used by some plants. We're not sure which. Some makes it a little bit deeper. And if there's any left over, it goes into deep water recharge. So this is thought to be important for competition. So it's long been thought that some species have shallower rooting systems, some have deeper, and so this can explain coexistence among species. Whether you have aspen and fir, are they using the same water, or are they effectively using different resources, grasses and shrubs versus trees? In order to understand this competition and niche partitioning, you have to know where the plants are getting their water from. And with climate change, it's going to be particularly important to have some idea of where plants are getting their water from. There's a pretty pervasive prediction that precipitation events will become fewer and larger. Right now there's relatively a lot of small events, but as the atmosphere warms, it can hold more moisture. It won't rain as often, but when it does, it'll rain more heavily. And how will that affect water cycling, energy exchange between the biosphere and the atmosphere, which affects climate, competition between different plants, and deep water infiltration. So if you want to know if there is going to be an increase in deep water infiltration, aquifer recharge, and water availability, you have to have some idea of where plants are getting their water from. Also tied in to fire. I think there's a growing appreciation for the fact that duff moisture and shallow soil moisture is critical for predicting fire intensity and timing and frequency. But it's really hard to predict how much water there will be at certain soil depths if you don't know where plants are pulling the water from. So, and there's one good reason why we don't know these things. It's a pain in the ass to work in soils. It's hard to dig a hole, right? Not only is it hard to dig a hole, but once you've dug the hole, you've probably killed everything you want to look at. On top of that, it's really these tiny little fine root hairs that are really hard to see that are actually doing all the heavy lifting of the water in the soil, and so it's hard to see those. And so my talk today is going to focus on this technique that we've been developing to try to figure out exactly where different plant species in the field get their water. But first, a little background. What do we know? The state of the art is well over 100 years old. You dig a hole and you make a picture of the roots. The problem is the roots that you see are these large roots, suberized roots that aren't really taking up the water. They're moving water up the plant, but they're not actually removing water from the soil. And so we don't really know the relationship between where root mass is and where plants get their water from. You can instrument soils with soil moisture sensors to get an idea of how water moves through the soil profile. And you can get really great measurements of the water cycle. The problem is you don't know which species are pulling the water from different depths and how climate change would affect competition among those species, or if you get a shift in species, how that will affect water cycling in the future. You can similarly use like a weir at a watershed scale to get an idea of how water moves through the whole system. But again, this doesn't give you information on how individual species access water. And like I mentioned, the trick is that it's really these tiny little microscopic root hairs that may only live for a few days that are really pulling all the water out of the soil. They're hard to see, almost impossible to distinguish different species. And so we have to find some way to figure out what these fine little root hairs are doing. There have been some techniques in the past 10 or 15 years that have come to the fore. Rhizotron systems where you look at roots, but again, you see them, but you don't know what they're taking up. You can look at water potential or stable isotope approaches to compare the isotope composition of plants and soils. But again, all these give you really fuzzy ideas. You can say, oh, this plant uses shallow water and that one uses some deeper water, but you don't really know really good quantitative ideas of where each species is getting the water. So we have been, at the point over here, we've been attacking this question. I started doing this work with invasive and native plants in the shrub steppe in Washington State. And then we really kicked into gear looking at this question of the two-layer hypothesis and tree and grass coexistence in Savannah systems. This work we've been doing mostly in Kruger Park in South Africa. So there's this long-standing hypothesis that trees and grasses can coexist in savannas because trees have access to deep soil water. The problem is when you measure roots, so the idea is trees have deep roots, grasses have shallow roots. The problem is when you measure roots, most of the roots are near the surface. This root biomass data doesn't really give us the answer. And after 50 years of this hypothesis explaining tree and grass coexistence, the data is lacking, and there's been a shift to the idea that, well, trees and grasses could probably coexist just because of fire and herbivory. And so these other explanations have become dominant explanations for why trees and grasses coexist, largely because we just don't have the data for water use. So we don't know if trees and grasses are using different soil resources. So our approach was a pulse chase approach where we inject deuterium oxide, a stable isotope of water, into the soil at several different soil depths a few times during the year, and then we follow that tracer, move through different plants. Consistent with the two-layer hypothesis, you'd expect that if you injected deep into the soil, you'd get pretty much a tree signal and no grass signal. And if you inject shallowly, you get mostly a grass signal with no tree signal. Alternatively, if there isn't niche partitioning, if you inject shallowly into the soil, you'll get both tree and grass water uptake. And if you inject deeply, you won't get much of a signal from any plant, right? So pretty simple idea. And again, what people would think going into this research is, well, plants get their water according to the root distribution. So this is the root biomass by depth. You get this nice power function decline with soil depth. So this would sort of be the default prediction of where trees and grasses get their water. When we inject the tracers, that's just what we see for grasses. It's like pretty beautiful. Grasses really get most of their water right from the surface. This isn't a semi-arid system. This data is from a site with 750 mils of precip. It's this woodland savanna system. Grasses really rely on this shallow water with less and less water uptake. They do get some water down below a meter, but not very much. And this is a pattern that we see year to year in a wide range of sites. And when we look at the trees, though, we see a slightly different pattern. And this is pretty nice results because it helps resolve these two long-standing conflicting perspectives on tree and grass water use. One, that all plants rely on shallow water, and two, the idea that trees rely on deeper water. What we see is that trees rely on slightly deeper water than grasses. All plants get most of their water from the top 20 centimeters of the soil, pretty small amounts of uptake from below 20 or 30 centimeters. But you see this niche partitioning at very shallow soil depths. This would be really hard to pick out just by looking at root biomass. So this approach has been working for us and giving us some pretty satisfying data. We've done this now. A nice thing about Kruger Park, other than the commute to work every day, is that there's a nice setup in the park where there's a dry north with 450 mils of precip and a wet south with 750 mils of precip. There's clay soils on the east and sand soils on the west. So just within a couple hundred kilometers, we can get really broad abiotic inference space, and we always get the same answer. You get, this is a sandy, dry site, and you see grasses rely on the surface, trees get water just below them, nobody uses deep soil. Same pattern at all the sites. This is a dry clay site, a wet sand site, and a wet clay site. And you always see grasses rely on the shallowest water, trees rely on water slightly below that. We've done some of this work up in Shrub Steppe, up in Washington State. We just finished a site up in Idaho this year, and I'm starting to look to do some work across an elevational gradient right here in Cache Valley, from agricultural sites all the way up to forest sites. But again, you know, two different sides of the world, very different systems. Plants typically rely on this very shallow soil. See, these are weedy plants in a weedy plant community early in the season. Later in the community, they start to shift, and you can see these different plants. This is diffuse knapweed, relying more heavily on that deep soil water. These are the native plants. It's balsam root, lupin, and blue bunch wheatgrass, relying mostly on this shallow water, moving slightly deeper later in the season. And we can start to look at some new ways of looking at plants and getting an idea of their niche partitioning. We did an experiment with, we actually put in 3 tree species, but the baboons really liked to eat one of the tree species, so we were only left with 2 species. But we planted saplings, and we grew them in little bags for a few months, so they had like 15, 20 centimeter roots. Planted them, they grew for about a month. We came back a month later, and after about 1 month of growth in the field, the saplings here in this dotted line relied more on deeper soil water than either adult trees or grasses, and they're putting down roots down to 1 meter. And so this is something we've seen in the Great Basin with cheatgrass. Cheatgrass can put down roots to 1 meter. Plants don't seem to have trouble getting their roots down to 1 meter or 2 meters. Even a tiny little annual plant can grow roots really deeply and really quickly, but there's not that much of a competitive demand for them to do so. Nitrogen is near the surface, and water comes from the sky, right? So there's a strong demand to use this shallow resources. Another thing we saw in this study is that grasses always, the black line here, grasses always show the same pattern. Every month, they always rely on the shallowest. They're very inflexible rooting patterns. Woody plants are much trickier. Woody plants relied on the shallowest water here and the shallowest water here. This was a wet month. And then as the soil's dried out, you see woody plants shifting their water uptake down to 50, 60 centimeters. So woody plants have this dynamic rooting system. They can put out several flushes of fine roots every year. Grasses always do the same thing, always relying on the shallowest soils. Another nice thing we can get with the tracer approach is we can get a deal of lateral root spread. Grasses, we do occasionally pick up grasses that will have lateral suckers. They'll get water from 2, 3, 4, 5 meters away, but it's less than 1% of their water uptake. Woody plants go well beyond their crowns, have wide lateral rooting, so they'll go 4 or 5 meters outside their crowns to get water. This is hot off the presses. I just pulled this data together last week. We just did a dual nitrogen deuterium tracer to see if plant roots are getting their nitrogen and deuterium in the same place, if it's passive uptake or if there's active root nitrogen uptake. And it looks like it's pretty passive. Plants have roots, and where those roots are, that's where they're getting their nitrogen. Grasses, you see this typical power function decline in nitrogen uptake with depth. Trees, you see this spike at 20 centimeters with a real heavy nitrogen uptake at that slightly deeper depth. So this is pretty basic biology. How can we use this information? One application we're using this for is to try to predict how trees and grasses and shrubs will respond to climate change. So there's been a lot of research, precipitation manipulation experiments looking at increases or decreases in precipitation. But a much more pervasive and poorly tested prediction of climate change is that precipitation events will become fewer and larger. And so we did an experiment where we created fewer and larger precipitation events. So we made these 8 by 8 meter shelters. We collected 50% of the precip, and then we redeposited that precip as only large events. So we didn't change the total amount of precipitation in a year. We just collected it and then redeposited it as 1 centimeter events, which are moderately large events for this system, not very large. And the reason we did this is because almost anything can happen when you change precipitation intensity. A lot of water, when you have many small events, a lot of water evaporates off the surface before it really helps plant transpiration. Fewer, larger events can decrease that interception and evaporation and actually increase water availability for all plants. But unless the storms become too big and they push water below the rooting zone, then you could decrease productivity of all plants, probably giving a competitive advantage to woody plants and increasing deep water infiltration. Or you could also get on steeper slopes, you could get some surface runoff. And so it really depends on exactly how much more intense your precipitation events become. And so we wanted to use an experiment to test this. Turned out to be tricky for us to figure out how to do this in a remote part of Africa. It took us quite a while to figure out the experimental design, but it ended up working. We collected water in these barrels and then distributed it through this tubing and ended up pushing water. So here's control soil moisture, plant available water down to 130 centimeters in control plots in blue and treated plots in red. And these treatments, just making these 1 centimeter events, pushed water deeply through the soil profile. So from our tracer studies, this would suggest that we're probably losing water from the soil profile, so this is bad for most plants, but probably better for trees than for grasses because there's a little bit slightly more deeper water. So we used dendrometers to measure woody plant growth increments, a disc pasture meter and harvests to measure grass biomass, and then we did a tracer study in the experiment to see how the root patterns responded. And consistent with our tracer data, this is woody plant diameter increments. The first couple years, there's no response by woody plants. But the second year of treatment, we see this big increase in woody plant growth. So as we would predict, we get this burst in woody plant growth with fewer larger precipitation events. So again, we didn't change the amount of precipitation, just the intensity of it. Grasses never liked it. Right from the beginning, we see less grass biomass in the treated than control plots. When we tried to get an idea of why this is happening, we looked at the proportion of tracer uptake in control and treated plots. Trees were relying on this surface soil with more deep water uptake than the grasses. And with treatments, they really shifted the rooting pattern. They killed off basically their shallow roots and moved their rooting pattern more deeply into the soil. Grasses looked like they're trying to do it, but they just weren't really able to shift their rooting pattern. So again, we see this inflexible grass rooting pattern and this flexible tree rooting pattern. So this helps explain how these woody plants are responding positively to this change in precipitation. So for this system, this is good for the browsers, good for giraffes and for elephants, not so good for impala and the lions that eat them, not so good for the white rhino. And a system like Intermountain West, this could help explain the shrub encroachment we see across much of the western U.S. And could help explain changes in fire regimes that result from shrub encroachment. So the next step for this work that we're just doing now is converting this proportional uptake. This has all been proportional uptake data that I've been talking about, which is good for looking at plant competition. But if you want to know the actual amount of water moving in and out of the system, you got to convert that to, you have to multiply the proportional uptake by transpiration, and that's work we're doing now. When you do that in this system, you see that grasses are really moving most of the water out of the system. This is a consistent grass carpet with occasional trees, so it's not too surprising. And we're getting some pretty promising results. We get R-squares of about 0.89. I can tell you how much water is, if you tell me the precipitation, I can tell you how much water is at any soil depth any day of the year. So this is pretty promising for modeling water, biosphere, atmosphere, energy exchange, deep water infiltration, and for predicting fire intensity at any given time. This is some work I'm also trying to now apply to the Intermountain West, looking at pinyon juniper responses to climate change, where we'll be tying my tracer data hopefully into a soil WAP model of water cycling and plant productivity. All right, and that's it. I have no idea how long that took, but I have to thank Andrew Mellon Foundation. They funded most of our work, all that work in Africa, as well as Utah Agricultural Experiment Station, USDA, and a number of other people. And the people who have injected over 500,000 holes into Kruger Park and injected all that deuterium. And that's it.
How would you predict that would things would go in areas with deeper water recharge with deep snowpacks, for example, like we have in our mountain systems?
Yeah. So this Savannah system has it's a different system in that it has a 6-month dry cold period, and then you get hot wet summers, and so you're always getting that precip from the surface down, and the plants use most of it before there's deep recharge, which is very different than the systems we have here, where we have a big snowpack that then infiltrates deeply, right? And so you see that in the Washington data, there was deeper water use, even though it's much smaller plants. You know, you get maybe blue bunch is a grass that grows this tall. The tropical grasses are 2+ meter tall grasses. They're huge grasses, but they only use 20, 30 centimeters. The smaller plants we have here go down 50, 60, 70 centimeters maybe. But still, it's not, nobody's getting much water from 70, 80 centimeters. There's not, most of the plant roots and water uptake is pretty shallow. There certainly are systems, it wasn't the case there, but there are systems like in the western US where trees will go to the aquifer, but it depends on the species and the depth to the water table. So yeah, but this is more of a plug that, you know, this is a new technique that we can now measure, and it's pretty fundamental. Like, it's amazing that we don't know where different plants get their water from. So this is something that I think we really need to apply to this region, and it's something that I'm starting to do, because it has the promise of allowing you to predict how species will respond to different climates. Yeah.
What role would a saltwater redistribution play in your experimental setup? Did you measure it? Was it happening?
So we inject and then we sample pretty much 2 days after. We have a pretty rough idea of the sap flux. So we know we're not getting redistributed water. We're getting the water that arrives in the plant root system pretty immediately. We know that the water that we measure is water that was taken up within a few hours of injection. So it's not measuring redistribution. We could measure redistribution a few different ways, but yeah, but our plants, it takes that long for the water to move up to our sampling sites. There's kind of, well, because our data integrates the whole root system, so it takes a while for it to come across the root system and from depth. And then some of the trees we're sampling up at 2 meters before we get it. So we'll see a quick spike within like 3 to 6 hours if we happen to have a direct line from our injection to the sample point. But most of the tracer flow peaks out at 2 days later. But there's a lot of details. I'm happy to talk to you later. Yeah.
Can you talk a little bit about implications for, you talked about that this has progress or implications for predicting water modeling use. Can you talk a little bit about how this could affect wildfire predictions and modeling and relationships with that? You mentioned that.
Yeah, so as I know Jim spoke earlier, I don't know if he's still here, but yeah, I've been talking to him, and I think for I'm not a fire specialist, but it's my understanding that having predictions or model estimates of duff moisture levels are pretty key for fire spread and intensity, and in order to know those duff and shallow soil moisture concentrations over a landscape, you have to know when it rained, how much it rained. You have to know how quickly water moves through that soil type. And most water in the western U.S. Goes back out to the atmosphere through plants. So if you want to know what water is doing, you have to know where plants are getting their water from. There's a big difference if plants are getting their water from 10 centimeters or 1 meter in determining how much water is near the surface. Yeah.
Climate models, because the climate models needed to run for many years, and so they usually group the taken into different types. called plant functional types. And usually they have 9 types. For example, for grasses, they have 2 types of grasses. One is called C3 grasses, and the other type is called C4 grasses. And C4 grasses have very deep roots, and it is as expected that in the future climate, tropic forests may be replaced by C4 grasses because the C4 grasses have very deep roots. And you should, the grasses can only use very shallow moisture, right? Did you measure
so
that kind of
The
very deep?
C4, I'm not sure the rooting, because of what I do, I'm skeptical of rooting distributions, but C4 grasses are more water use efficient because of the C4 pathway. I'm not as confident about the root depth distribution for C4 grasses. We do have species data, so we know what each species is doing. I just group them as trees and grasses to simplify it. And all the African grasses were C4. So and they're all using very shallow water. But I do have all our data is collected by species. We can tell you what an individual plant, it's usually individual plant samples that we take. So we know by individual, by species, but I just group it by functional type to make it easier to tell the story.
Thank you.